mode.
I'm the Executive Director of the Cholangiocarcinoma Foundation. My name is Donna Mayer, and I'd like to welcome you today to the first in a series of webinars about the Foundation's Research Fellowship Program. First, I want to note that if you have a specific question, you can ask it at any time in the question box on the right side of your screen, and then I will ask it on your behalf. Just a little bit of background about the Research Fellowship Program. It was established in 2015, and it's aimed at supporting early career researchers who focus on basic and translational studies in cholangiocarcinoma. The goal is to raise awareness about cholangiocarcinoma and inspire innovative quality research. Collaboration is essential to the success of any research program, and the Foundation chooses its grant recipients very carefully to ensure they will build working relationships between researchers, institutions, industry, and share information, samples, and expertise with others in the field. We're pleased to welcome our first presenter, Daniela Sia, the recipient of a $50,000 named grant in memory of Andrea Marie Fouquet, who was the young mother of 4 when she was diagnosed with cholangiocarcinoma. Danielle has been recently appointed as an assistant professor in the Department of Liver Diseases at Icahn School of Medicine at Mount Sinai in New York. Her research focuses on implementation of precision medicine approaches in intrahepatic cholangiocarcinoma. I welcome Danielle today, and you begin your presentation.
Good afternoon, Donna, and thank you very much for your kind introduction, and thanks to the Cholangiocarcinoma Foundation for the invitation to participate in this webinar series, giving us the opportunity to share our experience research. As you said, in the next 15-20 minutes, I will try to summarize our recent discovery and present an overview of the open questions that we will try to answer during the upcoming year, thanks to the fellowship received by Cholangiocarcinoma Foundation. I cannot move my slide. Okay, sorry for that. So even if I'm not a medical doctor, but a PhD and we focus on translational research, I would like to dedicate a couple of slides to summarize the current treatment algorithm for intrahepatic cholangiocarcinoma. Intrahepatic cholangiocarcinoma represents the second most common liver cancer after hepatocellular carcinoma and overall accounts for less than 5% of all gastrointestinal tumors. Typically, the prognosis is quite poor, with resection representing the main treatment option. At more advanced stage, chemotherapy regimens are considered standard of practice. Recently, a panel of experts gathered together and proposed a treatment algorithm for intrahepatic cholangiocarcinoma, known also as the ILCA guidelines for intrahepatic cholangiocarcinoma. According to this algorithm, following diagnosis, patients at early stage, usually stage 1 and 2, which represents 30–40% of the patients, can be treated with surgical resection. Unfortunately, the majority of patients is diagnosed in more advanced stage and represents 60–70% of the cases. In the patients with resectable disease, the resection can be curative or non-curative. In the second case, it should be followed by adjuvant therapy. In the case of the unresectable disease with the patients more advanced at the stage, there is currently no approved standard of care, and the recommended style of practice includes local regional chemoembolization and chemotherapy. Currently, intrahepatic cholangiocarcinoma is considered an orphan disease with increasing incidence and poor prognosis. There is only marginal understanding of the molecular pathogenesis, and there are no available molecular therapies right now. Thus, there is a clear need for integrative genomic analysis studies combining genetic alteration with signaling pathways, which would then provide the rationale for personalized medicine and patient stratification using either molecular subclasses or genetic biomarkers. In terms of molecular subclasses, our group has recently proposed a molecular classification which identifies 2 different groups of patients with intrahepatic cholangiocarcinoma. As you can see here, we have 2 main groups. One is named proliferation, the other one is named inflammation. These 2 groups are quite different in terms of pathways and have different clinical pathological parameters. However, without entering into many details, besides molecular classification, another way to move towards a more personalized approach to this disease relies on the identification of novel genetic biomarkers. for drug sensitivity or resistance. During the past decades, there has been a growing effort dedicated to understanding the molecular alteration underlying the development of this disease. Several studies have reported presence of many genetic mutations, like here indicated in this table, but most important, they have identified the regulated pathways and drugs that can inhibit these pathways. Unfortunately, to date, the results obtained in several phase 2 trials evaluating these targeted therapies have been discouraging. However, new hope has come recently from next-generation sequencing technologies, which have been successfully applied in many solid cancer in the last years to identify novel oncogenic drivers. In particular, fusion proteins are important driver oncogenes involved in the pathogenesis of many cancers, and recent studies have reported dramatic responses by blocking these targets. This is here an example in lung cancer where the identification of a specific fusion protein has yielded and obtained survival benefit in patients carrying such alteration by treating them with a specific drug named crizotinib. Our group was interested in trying to identify similar drivers in cholangiocarcinoma. For this reason, we recently applied RNA sequencing technology to cholangiocarcinoma samples, and we tried to characterize possible novel drivers like fusion protein as the one that I showed you before. For this reason, we have recently performed a study that was aimed at identifying such genetic alteration and characterize the oncogenic potential, and most important, if such alteration may represent novel targets for more specific therapies. By applying these new technologies, we have identified a novel fusion protein. With fusion protein, I refer to genes that are normally separated on different chromosomes, but because of some specific oncogenic mechanisms, they are fused together, as you can see here. The one we have identified in cholangiocarcinoma is between FGFR2 and PPHL1. These are two different genes that are located on different chromosomes. FGFR2 is fibroblast growth factor receptor that is involved in many functions important for the cell, like proliferation and survival, and is located on chromosome 10, whereas PPHL1 is a different gene involved in epithelial differentiation and is located on chromosome 12. In the cancer cell, in many cholangiocarcinoma cases, we have identified that these two genes, because of a translocation of the DNA, they are fused together and they generate a protein with oncogenic properties. We have explored the mechanism responsible, and as I said before, these two genes are in normal cells separate and are coded on different chromosomes. In cancer cell, however, in cholangiocarcinoma in particular, they are fused together to generate a novel protein with oncogenic properties. Here in the figure, you can see on the left, you have a normal cell where the two genes are separate, whereas on the right, because of translocation on the DNA, the cancer cell presents these two genes fused together to produce a novel protein. In normal cells, a fibroblast growth factor receptor is activated only by the presence of the ligand. Upon binding of the ligand, the receptor dimerizes, is activated and induces activation of the pathway within the cell. In the cancer cell, however, because of the presence of this FGFR2 fusion, where in our case the FGFR2 was fused to PPHL1, this fusion is always active, and we have always activation of the pathway within the cell that induces proliferation of the cancer cell. We explored the function of this novel fusion protein in vitro, and while normal fibroblasts do not form colonies, when we overexpress the fusion protein FGFR2 PPHL1, the normal fibroblasts NIH53 were growing in colonies. However, when we added an FGFR inhibitor to the culture, the colonies disappeared. Also, we transfected cholangiocarcinoma cell line and induced overexpression of this fusion, and we observed that the cancer cell overexpressing the fusion were growing faster compared to the cancer cell without the fusion protein. Also, the cells expressing our fusion protein were forming a higher number of colonies, suggesting that they were more aggressive, and also they were migrating faster. We were interested in this fusion because currently there are many FGFR2 inhibitors specific for this receptor. And so we tested the efficacy of these receptors in our system in vitro. And here you have on the left cancer cells expressing FGFR2 PPHL1 while treated in presence of this inhibitor, higher concentration of the inhibitor, this was responding to the treatment. Also, the migration was completely suppressed when we treated these cells with the presence of a GFR inhibitor. We were interested in understanding how many patients with cholangiocarcinoma may harbor this fusion. For this reason, we screened a large cohort of human samples of patients obtained from patients resected for cholangiocarcinoma, and we identified that 16% of our patients were positive for this fusion. Here again, I show you an example. So while on the left you have a negative patients where the two genes are separate, on the right you have an example of a patient positive for FGFR2 PPHL1, and again the two genes that are normally separate are fused together to produce this novel fusion protein. While we were working on our project, other groups reported similar results. However, other partners have been reported always fused to FGFR2, and other FGFR2 fusions have been reported like FGFR2-BEAC or this other fusion by another group. And so we tried to characterize our cohort and to identify how many patients may harbor FGFR2-BEAC. And in our case, we found that 38% of the patients were positive for this fusion. When we combined the results, we observed that the two fusions were not mutually exclusive, and actually 45% of the patients in our cohort were positive for at least one FGFR2 fusion, either FGFR2-BEAC or FGFR2-PPHL1. Unfortunately, we were not able to screen for the other fusion because of the absence of a positive control. I mentioned before that we proposed a molecular classification that divides the cholangiocarcinoma population into groups. One group is called proliferation subclass and is more aggressive and has lower survival and higher rate of recurrence, and another one that is more benign that is called inflammation subclass that is characterized by enrichment of inflammatory pathways. When we looked at how our fusions clustered, we didn't observe any enrichment in any of the subclasses, and we have 16% of our FGFR2 PPHL1 fusion, and as I said before, 38% of the patients were positive for the other FGFR2-BEAC fusion. Overall, we had 45% of patients positive for at least one FGFR2 fusion that might be candidate to receive specific FGFR inhibitors. We also screened our cohort for other alterations like KRAS mutation in the gene KRAS. We observed a 10%, and there was a strong association between presence of the FGFR2 fusion and presence of KRAS mutation. We also had ADH1 and IDH2 mutation. These are important because currently IDH1 and IDH2 inhibitors are currently investigated in clinical trials. They accounted for 17% of the population. We also had BRAF mutation in 4%, mutation in other oncogenes like ARAF and epidermal growth factor receptor in 11% and 2%. And finally, high-level amplification was observed in 4% of the patients. Overall, if we count all of these, we have the 70% of the patients with cholangiocarcinoma harbor at least one of these molecular alterations and can receive a specific therapy. Conclusion of our study and the open new avenues for the future is that we identified a novel FGFR2 fusion in 16% of patients with cholangiocarcinoma. As I said before, we were not the only group finding this alteration, but other groups and other investigators have reported a similar rearrangement between FGFR2 and other genes. How we demonstrated in vitro that this FGFR2 fusion promote the proliferation of the cholangiocarcinoma cells, but most importantly, they can be inhibited by FGFR2 inhibitor like BJJ398, currently investigated in clinical trials. Most importantly, when we combine our findings with other molecular alterations previously reported in cholangiocarcinoma, we observed that at least 70% of the patients harbor one molecular alteration and they might respond to specific molecular therapies. So it's clear that in the picture that I showed you before, FGFR2 fusion may have an important role in the development of cholangiocarcinoma. Most of it represent the more important targets for therapies. You can see here, but however, we have many open questions because the discovery of FGFR2 fusion, if it's important, also needs to be further explored. We have many open issues, like for example, the different FGFR2 fusion, as I say, we identified FGFR2 PPHL1, but other groups identified FGFR2 fused to other partners. So the first question is like, do the different fusions have the same oncogenic potential? And most of it, all of them responded the same way to specific FGFR inhibitors. Also, as we know, to detect the presence of this fusion, we need to have access to the tissue, to the cancer of each patient, and in some cases, and in most of the cases, these patients have undetectable disease. For this reason, we are interested in identifying if we can detect the presence of this fusion in the plasma of patients with intrahepatic cholangiocarcinoma. This will represent a minimally invasive technique that will allow us to test the patient for presence of this fusion and then classify them if able to receive or not specific therapies. Also, we want to design a specific low-cost device for the screening not only of the fusions but also of molecular alterations that have specific targeted therapies in order to guide a more personalized molecular treatment. With the help of the Cholangiocarcinoma Foundation and thanks to the fellowship that I received this year, we will try to work on these 4 open questions and try to find an answer. Thank you very much for your attention, and thank you to the Cholangiocarcinoma Foundation for your support. Thank you.
Thank you so much, Dr. Sia. There are no questions at this time, but we are open to answering any questions at any point in the webinar, so we might get back to you with additional follow-up. Our next presenter is Dr. Katsuyuki Miyabe, the recipient of a $40,000 named grant in memory of 12-year-old Christopher J. Wilkie, who passed away in March of 2014. Dr. Miyabe is a visiting scientist in the Division of Gastroenterology and Hepatology at Mayo Clinic in Rochester, Minnesota. He was born in Japan, where he earned his MD and PhD in gastroenterology. His overall goal is to obtain additional training in the study of cholangiocarcinoma in order to contribute meaningfully to improving the available treatments and care of patients with cholangiocarcinoma. Dr. Miyabe, I turn the floor over to you now.
Okay. Can you see the slide? Thank you for doing that. Can you see my slides?
No, not yet. Not at this point.
Yep.
Okay. Yes, we can see it now.
Okay. Yeah, thank you for the nice introducing. And it is a great honor to have the opportunity to present our work at this webinar. And I appreciate the award of the Christopher J. Wilkie Memorial Research Fellowship to me. My name is Katsuyuki Miyabe. I am a researcher working with Dr. Louis Roberts in the Division of Gastroenterology and Hepatology at Mayo Clinic, Rochester, Minnesota. Today, I will present my project entitled Fibroblast Growth Factor Receptor FGFR family gene aberrations in cholangiocarcinoma. First, I would like to introduce myself and my reasons for doing research in cholangiocarcinoma. I obtained my MD degree from GT Medical University in Japan in 1997. During my specialty training, I learned that cholangiocarcinoma had a poor prognosis and few treatment options. Therefore, I enrolled in graduate school in Japan with the goal of improving diagnostic procedures for early detection and developing new treatment for this cancer. After obtaining the Doctor of Philosophy degree in 2013, I worked as an assistant professor in the university. During my graduate studies, I noticed that an enormous amount of innovative research and publications have come from Mayo Clinic researchers, and determined to go there to develop my research skills. Furthermore, my cousin developed stones in her bile ducts after liver surgery for congenital bile duct dilatation, which is a major risk factor for cholangiocarcinoma. I am therefore highly motivated to learn about cholangiocarcinoma in order to further advance research on the treatment of cholangiocarcinoma. Last year, I was to meet with Dr. Louis Roberts at a conference in Kyoto, Japan, and I was strongly inspired with his research and knowledge of cholangiocarcinoma. Dr. Roberts invited me to participate in his research lab as a postdoctoral researcher to conduct research on the genetics of cholangiocarcinoma. As a background, cholangiocarcinoma, which I will abbreviate as CCA for the remainder of the presentation, is a lethal bile duct cancer with suboptimal treatment outcomes. CCA is more prevalent in Asia. For example, it is the sixth most common cause of death from cancer in my country. CCA is subclassified based on the location of the tumor as intrahepatic CCA, perihilar CCA, or distal CCA. And these are the same slides of the previous presentation. So this slide shows a picture that describes the classification of CCA. The liver and bile duct and gallbladder and pancreas and small intestine are shown. The gallbladder is connected to the bile duct through the cystic duct. So CCA developing within the liver is called intrahepatic CCA. CCA located in the bile duct above the cystic duct is called perihilar CCA. And the CCA located in the bile duct below the cystic duct is called distal CCA. CCA frequently presents at the advanced stage at which the current treatments are only minimally effective. Surgical resection and liver transplantation represent the only curative treatment for CCA. Systemic therapy with gemcitabine and cisplatin has been established as the standard of care for patients with advanced disease. This therapy prolongs median survival by only 3.5 months from 8 to 12 months, achieving only short-term survival. Thus, new therapeutic agents that can prolong the survival of CCA patients are crucially needed. Here, I would like to explain the relationship between DNA, RNA, and the protein. In cells, DNA is a database of instructions. RNA is made from DNA and carries a specific code of instructions to the protein synthesis machinery. And proteins are synthesized or constructed using the information from RNA. Thus, if there is an abnormality in DNA, the RNA becomes abnormal as well, and an abnormal protein will be produced. The abnormal protein can then cause changes in the cells and the tissues that lead to development of cancer. Next, I would like to explain what fibroblast growth factor receptor FGFR is. 4 different genes encode the fibroblast growth factor receptor proteins FGFR 1, 2, 3, and 4. When fibroblast growth factor binds to an FGFR receptor, this activates a signaling system within the cell that causes the cell to undergo certain changes, including cell proliferation, angiogenesis, and survival. In a number of types of cancers, such as breast, bladder, esophageal, ovarian cancers, genetic changes in the FGFR gene have been found, which cause excessive signaling. This hyperactivity of the FGFR receptors contributes to abnormal cell growth and cancer. Recently, many gene alterations that cause cancer have been found by researchers. Human genes sometimes are damaged by outside stimuli, such as ultraviolet rays, X-rays, and chemical toxins. Cells try to repair their own damaged DNA. Sometimes the repairs are incorrect or incomplete. The resulting abnormal changes include gene amplification, which means that more of the specific gene is made than in normal cells, or mutation, which means that changes occur in the DNA that can result in continuous activation of receptors, even in the absence of the corresponding growth factors. One of the alterations is fusion, which is a hybrid gene formed from two previously separated genes. The cell with some specific fusion will cause more of the receptor to be made than in normal cells and develop the cancer. Recently, fusions which cause a CCA have been found and a potential target for molecular targeted therapies. These figures show the procedure called fluorescence inside hybridization, which detects a specific fusion. These are two different cells performed fish using break-apart probe of FGFR2 fusion. The figure and the picture on the left side shows attached signal of red and green signals in a nucleus, which shows a chromosome without FGFR2 fusion. On the other hand, the slide on the right side shows separated signal of red and green signals in the nucleus, which shows FGFR2 fusion. In the previous presentation, she used a different type of fish probes. This type is a great apart fish probe, so it's different. In addition, a study of 156 CCA patients from Mayo Clinic also identified those fusions in 8% of patients. Interestingly, patients whose tumors have FGFR2 fusions survived significantly longer than patients without FGFR2 fusion. Patients with a fusion survive 123 months, while patients without a fusion survive 37 months. This suggests some other factors or gene alterations may affect the survival of patients with CCA. Indeed, a clinical trial at Mayo Clinic showed patients received targeted molecular therapy for those fusions. Look at the picture of the slide. These patients have CCA with FGFR fusion, and the FGFR inhibitor was effective for them. As we can see, red arrows in patient 1 and green arrows in patient 2. Additionally, we also recently evaluated the efficacy of a couple of drugs called FGFR inhibitors, which are created to inhibit the development by blocking the effect of FGFR fusion genes. As we can see in slides, we performed an experiment using mice. Each mouse was implanted patient-derived CCA cells, PDX, which have a FGFR fusion. As a result, all three FGFR inhibitors inhibited growth of CCA cells compared to CCA cells without drugs. Moreover, there are differences of effectiveness among those drugs. The importance is none of them completely eradicated xenograft tumors, which strongly indicates some other factors may affect the efficacy of those drugs. I would like to summarize the background. Fusions of FGFR genes have been discovered in CCA. Patients with a fusion may be related with longer survival. FGFR inhibitors blocked growth of CCA cells. However, none of them completely eradicated tumors. Based on these studies, we hypothesized that patients with other FGFR gene alterations could have worse prognosis, and these alterations are important therapeutic targets in CCA. Therefore, we propose further research to characterize the FGFR alterations that are most responsible to treatment with specific FGFR inhibitors. Then, I would like to describe how to investigate our hypothesis. There are three aims in this project. First, we will identify novel FGFR family gene aberrations in CCA. We will use a way called RNA sequencing, which enable us to get plenty of genetic information. RNA from 100 patient tissues will be analyzed by RNA sequencing to discover specific FGFR gene alterations. After getting results, we will confirm those alterations by other reliable procedures to make it tight. Second, we will evaluate the effect of FGFR protein alterations on signaling pathways during development of CCA. Once we determine specific RNA of FGFR family in M1, we can measure level of altered FGFR proteins in CCA and evaluate the effect of FGFR protein alterations on downstream signaling proteins. These approaches will lead to clarify the specific proteins for cancer development. Finally, we will evaluate the effectiveness of specific FGFR inhibitors in cell lines and mouse models bearing FGFR alterations. In cell lines, we will develop CCA cell lines bearing the novel FGFR alterations we discover and examine the growth of tumors bearing novel FGFR alterations in the presence or absence of FGFR inhibitors. In mouse models, we will evaluate the growth of CCA cells with specific gene alterations implanted subcutaneously in nude mice. In addition, we will test the effect of different FGFR inhibitors on implanted CCA PDX and determine the effectiveness of targeting FGFR alterations in treatment of CCA. These approaches will enable us to identify promising targetable FGFR alterations and the specific drugs that are most effective against each type of gene alteration. I would like to summarize this presentation. The many current challenges in treatment of CCA patients identify this field as a contemporary frontier of medicine. Therefore, we would like to identify additional novel FGFR target alterations and to determine their relative sensitivity to different FGFR inhibitors. The result of our studies will allow us to personalize the therapeutic options for patients with CCA by selecting the specific FGFR inhibitor that is most likely to achieve a clinical effect and therefore enhance patient outcomes. I would like to thank the Cholangiocarcinoma Foundation for their funding support to this study. This award will encourage me to achieve results promptly and contribute to me to perform further research in the next stage. Thank you for your attention. I would be happy to answer any questions. Thank you. That's it.
Okay, I guess Donna has been disconnected or having some audio issues. My name is Rick Pollack. I'm with the foundation. It doesn't look like we have any questions, but what we're going to do is we'll post this webinar up on our website, and if you have any questions, you can send them in, and we will make sure that they get to our two doctors. We want to thank you both for presenting today. It's not easy to do, especially when there's no reaction from the audience because everybody's on mute, but we just want to thank you, and thank you for taking the time to be here, and thanks everybody for coming and attending our webinar. Thanks, you guys.
Thank you.
Thank
Bye.
you.
Okay, bye-bye.
Bye.